All right, welcome everyone. Let us look at what we call the Krebs cycle. The Krebs cycle follows a series of reactions that we have so far discussed. It follows the link reaction. Remember, we start with glycolysis, but in between glycolysis and Krebs cycle, there is what we call the link reaction that we have already covered. So now we are moving to what we call the Krebs cycle, which happens to have several names. We can call it the citric acid cycle. We can call it the tricarboxylic cycle. Okay, we keep saying that it is a cycle because eventually you will come back to the point where the reaction started. Okay, so that is what we are going to look at. We are going to look at the different reactions that happen, and we will see the end products of these reactions, and we'll see what those end products are actually used for. But also we'll see in terms of energy, how many molecules of ATP are actually generated. Okay, so for example, for every molecule of glucose that we have it entering into the Krebs cycle, how many times shall we have the Krebs cycle running? Okay, so for every molecule pyruvate, the one which is the one of the end products of glycolysis, how many times will Krebs cycle be running? Okay, so that's what we are going to discuss in this session. So Krebs cycle, here we are saying that it is the third stage of aerobic respiration. Basically, the reason as why we are saying that is because we are including the link reaction. But if you remove the link reaction, then we have three stages of cellular respiration. We have glycolysis, and now if we have presence of oxygen, then we move to Krebs cycle, and then we move to the electron transport chain, or what you can basically call electron transport system, or you can call that oxidative phosphorylation. Okay, so here we are using Krebs cycle, the one that you're going to use to describe this cycle, because the name is actually derived from the person who discovered it. Okay, and that is Krebs. So Krebs is a name. Where does it occur? It occurs in the mitochondrial matrix. Remember, for glycolysis, it happens in the cytosol. Okay, but for now Krebs cycle, it happens in the mitochondrial matrix. Okay, and it immediately follows the link reaction. Remember, the link reaction connects glycolysis and Krebs cycle. So what enters into the Krebs cycle is basically acetyl-CoA. But remember, you have pyruvate as one of the major products of glycolysis. So the link reaction basically involves conversion of that pyruvate into acetyl-CoA that it can enter into the Krebs cycle. So the Krebs cycle has a number of reactions. They're actually 8 in number, and these ones are actually catalyzed by enzymes. The ultimate end goal is that you have acetyl-CoA being oxidized. Okay, oxidation of acetyl-CoA. That is the primary goal of Krebs cycle. Remember, when you have glucose entering glycolytic reactions or undergoing glycolysis, you end up with the two molecules or two compounds of pyruvate. Okay, so that means that for every single glucose molecule that we are going to have two pyruvate molecules. So that means that for every citric cycle, we are moving with one product, one compound, and that is one acetyl-CoA. So one pyruvate molecule gives you one acetyl-CoA, and that means that that acetyl-CoA is the one that enters Krebs cycle. So that means that for every one molecule of glucose, we are going to have Krebs cycle running two times, and that's what we are meaning here, that we have 2 times of the cycle of glucose molecule. The other unique characteristic of Krebs cycle is that you will have regeneration of intermediates for each time. That means that you don't have to keep having inputs of new other molecules to be used in Krebs cycle. As we go on, as the cycle goes on, what you used earlier on is replenished or is regenerated. Okay, so ultimately you will have ATP being produced, but again it will also be used in Krebs cycle later. Okay, it will actually be used later on in the respiration and in the respiratory processes. Okay, and then we have you have reduced coenzymes. Remember, we are talking about two coenzymes that are involved in respiratory reactions. We have NADH, okay, and then you have the flavin adenine dinucleotide. Okay, the reduced form is where you have the H. So if you have H2 here, this means this is the reduced form, and this also means this is the reduced form. So this is nicotinamide adenine dinucleotide. So both of them are coenzymes. In these respiratory reactions, what they actually do is that they accept hydrogen ions, so they become reduced. That means that where they get these hydrogen ions, they actually become those compounds become oxidized, whereas these coenzymes become reduced. And then you also have carbon dioxide as one of the products of the Krebs cycle, and it is what we breathe out. So let us look at the 8 steps of the Krebs cycle. So the first one involves a reaction between a compound that has 2 carbon atoms and then a compound that has 4 carbon atoms to give you a compound that has 6 carbon atoms. So what are these compounds? You have acetyl-CoA. Okay, basically the acetyl group of acetyl-CoA is the one that enters into the Krebs cycle. So that acetyl group has 2 carbon atoms. It combines with a 4 carbon compound that we call oxaloacetate, and then we form citrate, which is a 6 carbon compound. So the first step of this Krebs cycle involves the formation of citrate, which is a 6. I think the name also suggests citrate, 6 carbon atoms. Okay, so the enzyme involved there is what we call citrate synthetase. citric synthetase is the enzyme there. So the next reactions involve dehydrogenation and rehydrogenation. Removal of water and then adding water again to form a compound that we call isocitrate. Okay, so the next step, which is the third step, involves decarboxylation and dehydrogenation. So, like the name suggests, decarboxylation, you're removing carbon and therefore forming carbon dioxide. Okay, and then dehydrogenation means you're removing hydrogen. Now, when you remove hydrogen, what is happening there? That compound where you're removing the hydrogen from becomes oxidized. Okay, and the ones that receives that hydrogen becomes reduced, and that is where we are having NADH. Okay, so two reactions: decarboxylation and dehydrogenation. So that means that, remember, we started with a 6-carbon compound. Now we are moving to a carbon compound that has 1 carbon atom less, and that compound now becomes alpha ketoglutarate, and it has 5 carbon atoms. And remember, since the carbon was cut off, the products that you're going to have carbon dioxide, and then so the dehydrogenation gives you the NADH. Okay, so the enzymes that are involved here are isocitrate dehydrogenase and then oxalosuccinate decarboxylase. So step number 4 is now oxidation of alpha ketoglutarate. And still with oxidation, removal of hydrogen atoms from a compound. And that means the compound gets oxidized. So this process is what we call oxidative decarboxylation. Oxidative decarboxylation. Still, you're going to have another carbon being removed, and still you're having carbon dioxide coming out, and then also NADH. So step number 5 now it involves the conversion of succinyl CoA to succinate, and this requires an enzyme called succinyl CoA synthetase. That means that you're removing the CoA, and then you have the phosphate replacing it, and that results into the formation of GTP. Okay, that GTP donates its phosphate to ADP and eventually forming the ATP. Okay, so this is where we are now seeing that ATP is being synthesized. Okay. And then stage number 6, you have oxidation of succinate to fumarate, and then still oxidation. Remember, it's about removal of of hydrogen. And in this case, we are having flavin adenine dinucleotide coming in. Previously, we have been talking about nicotinamide adenine dinucleotide. Now, it's the first time we are seeing flavin adenine dinucleotide coming in. It's the one that gets reduced. In this case, it accepts the hydrogen from succinate. Okay, so the other reactions, you're having hydration, that is addition of water. At this point, if you can see, at this point, addition of water, to fumarate to form malate. And then finally you have oxidation of malate to oxaloacetate. Okay, so now we can say that we are actually having the oxaloacetate that we started with. So this is where we are saying that this Krebs cycle regenerates the intermediates. Okay, that's quite important there. So in summary, what we are saying is that for every cycle of per molecule of acetyl-CoA, you have Krebs cycle running once. But because for every molecule of glucose you have two aerobic molecules that will eventually be converted into acetyl-CoA, that means that for every molecule of glucose you will have Krebs cycle running at trans. We have seen that we have coenzymes NADH, NAD, and then FAD being used, and in this case they get reduced. Okay, so for every cycle you have 3 NAD being reduced to 1 FAD being reduced, 1 ATP molecule being produced, and then 2 molecules of carbon dioxide which will expire actually is actually given out through the Krebs cycle. That means that if we look at the complete 1 molecule of glucose, then we are going to multiply everything here by 2. Okay, because Krebs cycle will run thrice. But since it runs once here per acetyl-CoA, this is what you get. Okay, so that means that the cycle will keep running as long as we get what we get from the link reaction. So the link reaction is that feeds into gives the materials or the intermediates that will enter into Krebs cycle. So what does this really mean? It means that the Krebs cycle, yes, generates energy. That's 1. 2 is that we get reduced coenzymes that will later on be used in the electron transport chain.